Our eyes and brains help us make sense of the visual world around us. This is the case for many animals too, but their eyes and brains, or visual systems, can look very different to our ones. This is often because their visual systems have evolved to make best use of the environments in which they live, which are sometimes very different to the environments in which we live. However, what we don't know is how visual systems vary between closely related species which, on face value, appear to occupy very similar habitats. Knowing this would tell us how adaptable visual systems are and the role they may play in maintaining biological diversity. So, to answer this question, we decided to study a diverse group of animals which live in a pretty diverse environment, the Neotropics. These are the Ithomini, a tribe of around 400 butterfly species that I've been working on for the majority of my PhD. Now for this study, we decided to hone in on 4 species from that tribe and study them in detail. Those are Greta morganae, Mechanitis polymnia, Methona confusa, and Titharea harmonia. The natural history of these butterflies are relatively well understood, which means we can make relatively well-informed predictions about how their visual systems may vary. For example, Greta tends to prefer living in more shaded areas of the forest. So could this have an impact on how its visual system has evolved compared to the other 3 species? To answer questions like these, we needed to take a variety of different approaches. Remember, visual systems consist of the eye, the visually processing parts of the brain, and all the molecular and physiological machinery that keep the eye and the brain ticking. We wanted to investigate as many parts of the Ithomine butterfly visual system as we possibly could. So many in fact that I've only really got time to cover some of my favourite findings here. But we started off small, by looking at their genes. Specifically, we were interested in a group of genes called opsin genes. Opsins code for proteins which make up the main visual pigments found within animal eyes, including ours. Visual pigments are sensitive to certain wavelengths of light, giving animals which have several the ability to see colour. Now, Ithomine butterflies, just like humans, have 3 visual pigments in their eyes, but unlike humans, the sensitivity of those is slightly shifted towards shorter wavelengths. For example, Ithomines have a visual pigment which is most sensitive in the ultraviolet part of the spectrum, which we humans are not able to detect. We examined the opsin genes which underlie these visual pigments to see how they varied between our 4 species. And our main finding was that selection seems to be acting less strongly on the ultraviolet sensitive opsin of Greta. That suggests that being able to perceive ultraviolet light is less advantageous to Greta compared to the other 3 species. Now, any professional sunbathers out there will know that you're significantly less exposed to ultraviolet radiation if you're in the shade. That's why wearing sun cream on the beach is so important, right? So if you're like Greta and prefer shaded forest, then it's highly likely that there's less ultraviolet light to see in those habitats. This is really exciting because it provides the first evidence that visual systems can vary at the genetic level within these butterflies. But how might this scale up? Our next step was to see whether similar variation exists at the level of eye physiology and anatomy. In a previous video, I showed how we can make use of a remarkable property of butterfly eyes called the eye shine to understand how butterfly eyes physiologically respond to changes in their sensory world. And in another video, I showed how we can use micro-CT, a type of X-ray scanning, to acquire really detailed images of the internal workings of butterfly eyes and brains. Now, these data yielded a ton of results, but the overall message is that physiology and anatomy varied enormously between our species, providing further evidence that evolution has fine-tuned visual systems for the environments in which they're in. Just as an example, we showed that the eyes and visual brain regions of Greta to be relatively larger compared to the other 3 species, suggesting that these are an adaptation to the shaded selected forest habitat in which Greta lives for optimizing sensitivity. Now, we should emphasize that these are just hypotheses at this stage. More rigorous comparisons of the sensory ecology and behavior of these butterflies are necessary for us to properly unpick the evolutionary forces at play. But nevertheless, this provides pretty neat evidence that visual systems can vary at such a fine scale within a forest habitat, which is something we didn't really know before. I'm going to end with a final result from our study, which I think is particularly interesting, if a bit mysterious. It's also a great example of the unexpected discoveries you can make, which can kind of catch you a bit off guard when doing science. Now, so far, I've only spoken about differences between our four species. But what about differences between males and females within the same species? When perusing our butterfly brain scans, we stumbled across a brain region called the ventral lobula, which is present in females, but entirely absent in males of some species. Now, in other butterflies, we know the ventral lobula is involved in processing visual information coming from the eye, but our research suggests that the way it's doing that is slightly different for ethomine butterflies. Now, we can assume in ethomines that the ventral lobula is controlling behaviours which are specific to females. But to be honest, the rest is one big guessing game. A lot more research is needed to properly explore this mystery, but for now it provides a striking example of just how dramatically visual systems can vary, even within the same species. So that was a very brief whistle-stop tour of our most recent paper, which is now published in the Journal of Experimental Biology. If you want to find out more, then I've posted a link to the full paper in the description of this video. For now, I hope you enjoyed this. If you did, then don't forget to subscribe to the channel to stay updated with all my wildlife and science adventures. In the meantime, I'll see you next time.